Hello and welcome to this module 2 of tissues, looking at connective tissue and its classification. This is the first part of 3 parts of this connective tissue online lecture, looking at the introduction to connective tissue. So the learning objectives for this first part of the recording is to compare and contrast the structure, function and location of connective tissue with the other 3 primary tissue types. Now this is an extended learning objective, meaning that it not only extends to today's recording but also to the rest of the tissue lectures that you've looked at. So the previous epithelial tissue online modules, as well as the lectures which you'll have on muscle and nervous tissue. So it's quite an extended learning objective over the course of this semester. And you should be able to describe the structure and function of ordinary connective tissue, including its cells and extracellular matrix. So let's get started. So what is connective tissue? Well, this seems like a really dumb question, but connective tissue is not epithelium, it's not muscle, and it's not nerve. So if it's not those 3 basic building block tissues, then it must be everything else in between. And it just seems a bit of a dumb thing to say, but in fact, without connective tissue, epithelium, muscle and nervous tissue wouldn't function because they all rely on connective tissue. So whenever students start to learn about tissues, they quite often ignore connective tissue because, it's not as fabulous as epithelium and it's not as dramatic as muscle, and certainly we don't think of it as being as important as nervous tissue. But as I say, without connective tissue, epithelium can't get its blood supply, so it's not going to live. Muscle tissue can't contract against anything, so it can't perform its function. And nervous tissue can't get its blood supply either because blood vessels, nerves, lymphatics all use connective tissue to basically allow nervous tissue and these other tissues to function. So connective tissue forms our skeleton, so our skeletal system, so bone and cartilage is a type of connective tissue. Blood and blood-forming tissues is a type of connective tissue. You may not think about that very often, but in fact, blood is actually a type of connective tissue. Obviously, very liquid type of connective tissue, and on the opposite scale to bone tissue. If you think bone tissue is very hard substance, blood being liquid, obviously they're on opposite ends of the scale of the structure of connective tissue types. Fat, adipose tissue, which we'll be talking in the last part of the module today, is a type of connective tissue. Ligaments and tendons, which join bones and muscles to bones, important component to enabling muscle to work, for example, to enable contraction and movement of joints. And the supporting tissue. So again, this idea that connective tissue is a really supporting tissue for these other tissue types. So it supports the other tissues. So those first 3 types of connective tissue, the skeleton, blood and fat, are all what we call special connective tissue. Whereas the ligaments and tendons and the stroma, the supporting tissue, is what we define as ordinary connective tissue. So this is sort of a classification of connective tissue. It's either ordinary connective tissue or it's special connective tissue. So the purpose of today is to learn a little bit about particularly ordinary connective tissue and then finish off with a little example of special connective tissue in the form of adipose tissue. So in the previous online module, we had a look at epithelium and we saw that if there's a surface, there's a lining of epithelium associated with it. And so here we've got all the common examples which we find in the human body. So here we have 8 different types. So we've got the simple squamous endothelium, lining the blood vessels, so it's a simple squamous epithelium. We have the simple cuboidal in the kidney tubules. We have a simple ciliated columnar in the uterine tube. We have our pseudostratified ciliated columnar in the respiratory tract, in the trachea. We have the stratified squamous non-keratinized with the nuclei all the way to the surface in the vagina. Stratified squamous keratinized, which is an example of the epidermis of the skin. Stratified columnar of the salivary gland, and the transitional epithelium, that real unique type of epithelium which enables expansion within the urinary system. Now, the connective tissue is quite difficult to see in the simple squamous and in the simple cuboidal. But if we look at the other tissue types, the connective tissue is really a prominent feature of these pictures here. So if we have a look, uterine tube, there's the connective tissue. The trachea, there's the connective tissue. Here's the vagina, there's the connective tissue. Stratified squamous in the skin. in the duct of the salivary gland, and if we look at the ureter there, all of that is connective tissue. Remembering that one of the key features of epithelium is that every epithelium has an underlying layer of connective tissue beneath it. So if we compare them side by side, we'll find that epithelium and connective tissue are really quite different from each other. Epithelium has many cells. It was characterized by having lots and lots of cells very closely adhered together, whereas connective tissue actually has few cells in comparison. Epithelium has those very densely packed cells, whereas connective tissue, the cells are actually quite scattered, quite separate from each other in most cases. Epithelium, because of those densely packed cells, what that means is that there's very little evidence of extracellular material or extracellular space between those cells. All the cells are really tightly adhered together, whereas the opposite happens in connective tissue. Most of connective tissue, funnily enough, is extracellular matrix. And there's a few cells scattered around, so it's very different to epithelium there. And epithelial tissue formed the tissue by having lots of different types of cell junctions, so tight junctions and adhering junctions, which enabled all the epithelial cells to stick together and form their own type of tissue, the epithelial tissue. Whereas there's no contact or junctions between cells. Now, I've put in brackets here usually because there is one example, and that's the osteocytes, which are the bone cells, which actually do have connective tissue. They do have junctions between them, but other than that, there's no other junctions between connective tissue cells. So remember the two ways that tissues can form, either through cell junctions and or extracellular matrix. Epithelium forms a tissue through its cell junctions. Connective tissue forms its tissue through the extracellular matrix, binding it all together. So here we have the two opposite poles, cell junctions, extracellular matrix. So here we've got our section of skin. So this is a stratified squamous keratinized epithelium, and you can see all the different cell types within the stratified squamous epithelium. There's no nuclei at the top. That's a dead layer of keratinized cells at the top there, and directly beneath it we actually have the connective tissue. And in fact, there's two different types of connective tissue there, of which we classify them as ordinary connective tissue. And you'll see a few things happening in here. So you might be able to see a few blood vessels. Those little red blood cells there, those are some capillaries and some small blood vessels there. There's some lymphatics present. So all blood vessels, lymphatics, nerves quite often use connective tissue to get to their where they're going. So what actually makes up connective tissue? So we know epithelium is made up of cells. That's pretty much all they're made up of, with a few junctions and things like that. But what about connective tissue? Because we know we've got all this extracellular matrix stuff going on. What's going on? So connective tissue is made up of cells. And the main cell type within connective tissue are the connective tissue cells, which we call fibroblasts. Fibro relating to fibres, which is part of the extracellular matrix, and blast is a term which we refer to as sort of an immature cell or an actively synthesizing cell. So fibroblasts are actively synthesizing the extracellular matrix. We find other cells within connective tissue because, as I said at the beginning, connective tissue is quite often taken over by other components, so blood vessels, nerves, lymphatics, and in fact the immune system is a big component of connective tissue, or may be a big component of connective tissue. So we find macrophages, which are the little resident phagocytes which can engulf anything which has bypassed the epithelium and gone into the connective tissue. Mast cells are resident cells within there which can initiate immune responses and inflammatory responses. Plasma cells, again, they can produce immunoglobulins or antibodies, again, part of the immune system. And then we have a number of other white blood cells. So in fact, when we have a look at what makes up the cells of connective tissue, we can sort of divide them into fibroblasts and then other cells, and those other cells are what we refer to as fixed cells. So they're almost always there, present within the extracellular matrix, so the macrophages, the mast cells, the plasma cells. And then we have other cells which are called transient cells. They come and go depending upon the needs of that tissue. So if there's been a bacteria come in, then we can get infiltration by these white blood cells. We can get neutrophils coming to phagocytose stuff. We can get lymphocytes coming to get the antigen and take it to a lymph node to initiate an immune response and so on. So we have fixed cells and transient cells. The second component of connective tissue, of course, is the big component of connective tissue, and that's what defines the structure and function of connective tissue, and that's the extracellular matrix. And the extracellular matrix is made up of ground substance, which is sometimes referred to as amorphous ground substance. Amorphous just means without shape. So morph referring to shape. When you put A in front of something, it just means without. So it's amorphous. Like avascular means without blood supply, which we talked about with epithelium. And ground substance is made up of glycosaminoglycans, which we just call GAGs, and proteoglycans, and it's also made up of water. And those amounts may vary depending upon the needs of that connective tissue. The other component of the extracellular matrix is fibres, and we find sort of 2 to 3 different types of fibres. The most abundant fibre in the human body, the most abundant protein in the human body is collagen. So collagen is the most abundant connective tissue fibre, and it's secreted by the fibroblasts. So all of the extracellular matrix is secreted by the fibroblasts. There's a second type called reticular fibres, and these are a special type of collagen fibre, sort of small, thin collagen fibres, which support a lot of the glands and also a lot of the lymphatic tissues and things. And then elastic fibres, which are an important component of some of our blood vessels and also our skin. It gives elasticity to whatever organ they're supplying. So connective tissue, when I was a student, one of the things I really enjoyed was when things made sense, very logical sense. And so I've come up with my own rule of twos for connective tissue. It's not entirely perfect, but it's a reasonable way to remember the structure of connective tissue. And so this is my own version of the way to try to remember connective tissue by using a sort of a flow diagram. Now, I presented this in a lecture last year, so what I'm going to do is I'm going to cut and paste this little section from last year's lecture where I drew the rule of twos into this video, and then we'll move on. Okay, connective tissue is made up of 2 components. Cells and extracellular matrix. So there's our first 2. Connective tissue is made up of cells and extracellular matrix. The extracellular matrix is made up of ground substance, and fibres. Ground substance is made up of GAGs and water. Water is made up of hydrogen and oxygen. No, I don't got to go to that level, but you get the idea. Fibres are made up of collagen and elastin, or elastic fibres. Now, there was that third type, reticular fibres. They were initially described as being different, but in fact, when they went to the biochemistry, they saw that reticular fibres were one type of collagen fibres. Okay, so reticular fibres are collagen fibres. So there's our 2 again. Cells fibroblasts, and then other cells, it's not that easy to see that, is it? Fixed cells and what we call mobile cells, or cells which are transient. They sort of come in and they come out depending upon their need within that tissue. So we know that the fixed cells include things like macrophages and plasma cells and things like that. The mobile ones are the things like the lymphocytes and the neutrophils and the basophils, any of the white blood cells that can move around within that tissue. So this is the rule of two that I've sort of come up with for connective tissue. So here we have the example of the different types of connective tissues. Here, particularly loose connective tissue may have all these different cell types. So we have our fixed cells. Here they've listed melanocytes, but we talked about macrophages. We talked about mast cells. We talked about fibroblasts being the main cells which secrete the extracellular matrix and things like that. And then we have our wandering cells. Plasma cells and lymphocytes and other white blood cells. We may find adipocytes present there as well. So fibroblasts are the true connective tissue cells. They're what are responsible for creating the extracellular matrix, so the ground substance and the fibers. So both of those components are made by fibroblasts. So when we use the suffix blasts, it refers to an immature or an active cell. So it's an early cell which is synthesizing extracellular matrix. So it's making the extracellular matrix, the ground substance and the fibers. And it's characterized by being quite a relatively large amount of cytoplasm, and within that cytoplasm we have lots of rough endoplasmic reticulum, we have lots of Golgi, and so on, organelles which are basically producing these proteins in the form of collagens and glycosaminoglycans and elastins, and then exocytosing them out into the extracellular space. Now, when fibroblasts have completed their job, they sort of become quiescent. That means that they sort of quieten down and they form fibrocytes. So fibrocytes, the suffix "cytes" refers to cell or mature cell. So fibrocytes are relatively inactive. They contain little amounts of rough endoplasmic reticulum and Golgi because there's not a lot of synthesis going on of these extracellular matrix components. But that doesn't mean that they can't revert back into a fibroblast-like state where they synthesize more extracellular matrix. So fibrocytes are responsible for maintenance of connective tissue, whereas fibroblasts are responsible for synthesis of extracellular matrix in connective tissue. So fibrocytes, if something happens and we need to make more extracellular matrix, fibrocytes can revert back to a fibroblast-like state and synthesize new extracellular matrix. So either way, fibroblasts are the actively synthesizing cells producing extracellular matrix. Fibrocytes are about maintaining the extracellular matrix, so they'll make sure everything's okay. So with histology, we actually use special types of stains to identify connective tissue, because for the most part, ground substance gets removed when we process for microscopy. Collagen fibers aren't particularly good at being stained with hematoxylin and eosin. So in order to display connective tissue, we can use special stains. So a common stain which we'll use when we are particularly interested in connective tissue is a stain called a trichrome stain, which means 3 different types of colours. And when we use a trichrome stain, collagen's usually stained blue or green, and then the other components are stained red and yellow and things like that. If we want to visualise elastic fibers, then we'll use a special stain called Aucian, and that tends to stain elastic fibers black or dark red or magenta there. Or of course we can go to the electron microscope, and in the little right hand corner down here you can see some collagen fibers in longitudinal and in cross section. At the top right is the cross section, and in longitudinal down the bottom. And collagen typically has this sort of striated appearance where it's got these bands, and that's relating to the way in which the proteins sort of fold upon each other. So fibroblasts make the collagen protein of the collagenous fibers, and they use amino acids to put these all together. And collagen is actually made up of a triple helix. So DNA was made up of a double helix with 2 bands wrapping together. Collagen is actually sort of the molecular equivalent of a rope, where all of those collagen proteins are tied together really tightly in this triple helix. And it's an extraordinarily strong protein that in the equivalent of the world, it's one of the strongest things which we have available to us. So our bodies are made up of this very strong connective tissue protein in the form of collagen. So down on the bottom here, we've got our fibroblasts with lots of rough endoplasmic reticulum, big expanded cisternae, and then outside in the extracellular space, we have our collagen fibrils, and you can see the striations which we were talking about before. On the right-hand side, we've got a H&E stained section. The arrows are pointing to the nuclei of fibroblasts. They're quite round, indicating they're likely fibroblasts. The more flattened ones are getting close to being fibrocytes, but we've got all this extracellular matrix in between. All those white spaces, that would be the where the ground substance was. Okay, so that would all be expanding within there, but as I say, when we process for microscopy, quite often the ground substance is removed. So whenever you see a micrograph like this, pink, fibers, you can see the big bundles of fibers. If you've got white spaces in between, you should be thinking in the native form there would be ground substance between there. And ground substance, because it's largely water and glycosaminoglycans, it allows diffusion to happen. So collagen fibers actually form extracellularly. So the fibroblasts produce what's called the procollagen, or the immature collagen. They secrete it into the extracellular space. It gets modified by enzymes into its final form as a collagen fibril, and then it gets bundled together to form a collagen fiber. Now, one thing that's really important for this process is actually a vitamin, and that vitamin is vitamin C. If you don't have enough vitamin C, you end up with something called scurvy. And in people who suffer from scurvy, one of the things that happens is their teeth fall out, because in your teeth, what holds them to the bone are little periodontal membranes, connective tissue, which holds your teeth in place within the sockets within your jaw. And without collagen, you don't renew this connective tissue, and so your teeth become loose and they may fall out. And same with the hair, the connective tissue holds sort of their hair in place, and so people with scurvy tend to have hair falling out. They tend to bleed a lot from the gums, and their teeth fall out. So the people who used to go on long voyages, particularly the British when they were first colonising the world, one of the things that they used to carry with them were limes. And limes are a great source of vitamin C, and so they take these long voyages, they suck on a bit of lime, they get their vitamin C, and then they wouldn't end up with scurvy. And so Americans actually call British sometimes limeys, because of the fact that they were trying to prevent scurvy by sucking on limes and having the limes present on board for long voyages to colonise the world. Just a little bit funny fact there. So collagen can exist as either fine fibrils or very coarse bundles, depending on the type of collagen and what other proteins are present. So there are different types of connective tissue which require fine fibrils, and there are other types of connective tissue which require strength, and therefore the collagen exists as these big bundles. And we'll talk about those in the next section. So there's a whole family of collagen proteins that are similar, but they have different arrangements, different distributions within the body, they have different functions, and they're made up of different types of collagen. And in fact, the human body, so far we've identified around about 30 or more different types of collagen. There are these common types of collagen. These are the most common types of collagen. So type 1 collagen we find within ordinary connective tissue and bone. Type 1 collagen was the first to be discovered and to be characterized, and it's the most abundant in the human body. So type 1 collagen is the most abundant collagen. Type 2 collagen we find specifically in cartilage. Type 3 collagen, these are reticular fibers. So remember, we have those three types of fibers: collagen, reticular, elastic fibers. Well, type 3 collagen is a specific type of fiber, which is the reticular fiber. Type 4 we find in the basal lamina, so we talked about that as part of epithelial tissue. Type 5 we find in muscle and tendon sheaths, and almost 30, as I say, different types of collagen have been described so far. So just keep in mind that fibroblasts, of course, not only make collagen, but they also make the elastic fibers and the ground substance, so the gags, and the gags attract water. So fibroblasts secrete elastin in a sort of a proelastin form that associates with other proteins, and then they form these elastic fibers. And so they're, again, important components of our connective tissue. So this ends part 1 of the module. So what you'll find in the next section is a short quiz just to test your understanding before you move on to the video relating to part 2. So I'll see you in part 2 of this video module.